So good afternoon here in 502 and also those who are on the webinar. It's a great pleasure to introduce in this series of the Department of Epidemiology seminars Dr. Jaap Goudsmit. Jaap Goudsmit is an adjunct professor in epidemiology and in infectious disease and immunology. Originally was at the University of Amsterdam, virology department. Went into industry with Johnson & Johnson, in particular Janssen's vaccine company, and then since 2017 joined us here as well. So it's a great pleasure, Jaap, to have you and to talk about pandemic preparedness, the role of virus-restricted vaccines and universal antiviral drugs. Please, Dr. Goudsmit.
So, everybody, welcome. Thank you for joining me in this endeavor. And what I want to talk about in this, say, 3/4 of an hour is a little bit the time frame we are in and what can we do, because pandemic preparedness is a very complicated issue in terms of we don't know what's coming. And I try to show you a set of lessons I learned about the past. What have I seen happening and what goes wrong in what you see happening? So it is a little bit a kind of asking yourself, can you prepare yourself for a pandemic? And if so, how would you go about it? So the first thing is, my first topic is, it starts out with is it really true that when a virus enters multiple species during all the seasons, is that actually the first sign that something also gets being transmitted from human to human? Is that actually true? And the biggest issue there is that yearly people think that about 50 to 100 times there are infections, zoonotic viral infections occurring, but they generally infect either no person at all or only one generation of persons, and then it stops. So the key question then is, what is the moment that it really hits off in the human population? And what have we learned from previous experiences? So what can we say, or what are people saying about the next pandemic? And the big issue is a little bit, can we say anything about what's coming next? And there are two views out there. One of the views is that it can happen anytime now, soon. So we have to be very prepared for this earliest moment. And the biggest problem, and I'm somewhat older, so I've seen a lot of this kind of thinking, but there is another school now, which was exemplified by this paper in PNAS, which suggested that you have to look more to the severity of the pandemic and to certain characteristics which defines that severity. If you do that, they came up with this interesting, and I like this paper a lot, that says it looks like that everybody is thinking in what they experience themselves is what's going to happen multiple times after that again. In other words, we are all experiencing, and most of us, the pandemic, COVID pandemic and all his fallout of that. And is that then a measure of that it will occur the coming few years again? The problem with preparing yourself for pandemics, I can tell you the funding cycle after SARS or the 1976 swine flu, so-called pandemic, and give us a little view on what happens. So what these guys in this PNAS paper suggested is, if you look at severity as well, what you see is that actually there are maybe only once in a generation this kind of pandemics. And then they compare only the pandemic of COVID to the 1918 flu. And they say, listen, now we're talking, those are the ones you have to prepare yourself for if you can. Now, that means that's very rare. Now, that's an interesting view, and I will go through that reasoning a little bit. Now, the issue is we see a hell of a lot of infections occurring, particularly of certain viruses, which I will discuss, how you can narrow what might be coming, although we don't know when. And one of the ones that is very popular now is, of course, H5N1, which is a flu strain. It's a flu strain which originally comes from bats. That flu strain then subsequently went into a lot of other animals. It was in poultry, subsequently also in a lot of other animals. And the last few years, it ended up in almost every mammal that you can imagine year-round. So you again have a kind of question, is a panzoonotic infection really hitting off, yes or no, and are there parameters for that? At the same time, as I said already, several other ones showed up, like a bat virus, which was a coronavirus like MERS. I will talk a little bit about that history. There are other flu strains which showed up in different countries in the world. So the key question there is, can it happen every day? The biggest problem we have, and that is a kind of interesting one, is how the institutions like the WHO, but also the CDC and all the other ones, actually handle what is coming next. And they took what I call a very looking back effort in that. If you look at this slide, the WHO has first decided what are the priority diseases with the highest risk of showing up as a pandemic. But what you see in that list is merely infections that occurred but never actually created a pandemic. There were epidemics at certain locales. If you look at hemorrhagic fever by the Crimean Congo virus or Ebola in Marburg, any of those, it looks like they looked at severity particularly, but not at the spread of the severity problem. So, in my opinion, scientifically, that didn't help much. Subsequently, they changed in the years after. They changed to a tactic of looking at families, which I find already more elegant. But then the question is, can you, and we were just discussing a little bit, can you find either vaccines or drugs that actually are uniform enough that you can give them to everybody? The FDA, and particularly, the most interesting measures I see in organizations like of the Army, which is, for instance, BARDA, who says we want to see a few things. If you prepare yourself for a pandemic, we want to actually have something on the market already for the normal family of viruses. So you have a flu drug or a flu vaccine that is broad enough to include whatever pandemic strain is coming, which I find a more elegant way than to go to all kinds of families and finding the origin. And we go a little bit through that reasoning. Now, the most dramatic demonstration of how complex this issue is the last COVID pandemic. I remember when it was also during the course, which was then early in the year, so we're talking January here in our vaccine course for the first time. We were discussing it in January of that time. And there was a lot of politics around it. One is, where does it come from? Which I find quite irrelevant, because mistakes in the laboratory, I know from a lot of occasions that occurred, up to very dangerous ones, like in Holland we had an escape of polio out of a polio factory. I give you one example, but there were several other ones. They occur, of course, in some kind of regularity. Not that frequent, but they occur. So a lab escape is generally occurring at a lot of times. The biggest problem with that kind of thinking is, where do you put your fear, in a way? And my thinking is, as soon as you get the first cases of human-to-human spread, as I said before, this was interesting. When the first cases occurred in China, this was 2019, either in November or December or even a little earlier. When that first occurred, I had, I think about a quarter of the class were from China. And we changed the whole class, interestingly enough, into more or less a master class where they were telling what they heard in their own country, which was a kind of change we made pretty fast. And they were reporting every time, twice a week in the class, what they heard. So we knew a little bit how complex it was. So I tell you something, that one student was saying, yeah, my mother has cancer, and she was in a hospital, which was not even in Wuhan, but one of the other provinces. And at a certain point, no doctors were anymore at the hospital because they were all to Wuhan. So then you know something is going on. But then how you react on a local pandemic, and how serious do you take it? The next event that occurred was in Italy for Europe. And that next event was in one region where there was also a big fair for people who dealt with leather and shoes and all that. So that occurred there was dramatic. I mean, you saw coffins in the streets in Bergamo and all those places around it in a certain area of Italy. Then what occurred was then in southern Holland, in Brabant, a very small area, there showed that there was in one hospital, and its single source was a man which came from Atlanta from this fair. Even then, we had discussions with the government to say, listen, this might getting serious now. It was very difficult to apply certain measures which would have been logical, like when there were proposals on the table like asking the army to close off this province. Now, everybody was, of course, against it. And it never happened. It might have been logical. So it took a lot of effort to get it into your brain that you really have to start thinking. Now, if we go back from human-to-human transmission recognition, I wanted to show you this slide. SARS-CoV-2, the origin of all these viruses, there's a lot of effort, let us put it that way, in the world of surveilling particularly bats for coronaviruses at the time. There was already for 20 years an international effort to do that. If you look now, how relevant is it to know that one of the subset of viruses is actually doing something terrible? And what you see here is interestingly enough, they found about 10,000 different bat viruses in China and in the Philippines and in southern Asia. And there was one which was very close to the viruses that spread among humans. Now, a key question then is, this was 2000...
For purposes of people who listen to you, have to speak.
But I have...
you have one info. Sorry.
I have to put my microphone on. I'm sorry.
Yeah. And anyway, they see you then.
Yeah, they see me. Okay, great. So my worry a little bit about this is that we had 10,000 of these viruses out of bats. Which one will it be that actually shows up out of this whole phylogenetic tree of determining the different viruses in bats? And bats harbor the most virus families from any species. That is always amazing. There's a big discussion on why that is on immunological level. But that one bat virus was, of course, never identified earlier than 2020, when it was finally sequenced and put into this tree. So in other words, what's the use of making this enormous surveillance? And we can discuss that. And I think it is a terrible mistake to spend all that time on getting all the bat viruses in the world mapped. Now then, the key issue is, from which animal is it actually coming? We only know that generally they have to go through several mutations, these viruses, and these mutations generally occur from species to species transmission, particularly in mammals. And we know that from flu, that's what I learned in medical school early on, is it goes from why does it always come from China? Because they live together, these poultry as well as pigs as humans. So you see here just a picture of how you can approach this issue. However, the problem, of course, with all these problems is that still you don't know more than you originally knew, except it comes maybe from an animal. The next problem is, of course, the speed at which something spills out of these mammal species. Now, there is a lot of data that suggests that if you have higher intensity, as I mentioned just a second ago, of living together of different species of mammals with humans, is that a trigger for any future pandemic? And yes, it might be, no, it might not be, because it not always occurred, particularly if it's such rare events. like the 1918 flu. It was not the case. It was directly from poultry. And then with the COVID pandemic, we don't even know what the intermediate species was, despite the fact what everybody says. It might be, and that is different, and I will tell you in a second, in what we knew till that moment. That was the biggest problem. All the time, we're taking science as it is a reflection from the moment itself, and we are more wrong than right, in my opinion, but that's maybe because of my age. So one of the key questions is the change in climate actually speeding up the way that actually pandemics can occur or not? And we just don't know that as yet, but there is a lot of debate on that issue. Now, the nice thing is people try to grasp actually the speed and where it comes from and locale in this whole pandemic preparedness discussion. And there is one model I really liked, which is it was published in PNAS, and presumably the slides are available to everybody, so you can just look it up, and it is the best model at the moment, based on virological characteristics, and based on the pandemics that we thought, and they put a lot of data in there. And what you see is, and I just give it as one thought, is that the thought was that they included a set of criteria, which I show you in a second, to actually unify how you can maybe assess a little bit in a model what happened if you put all the data over the last 100 years in there, whatever we can. And the data they put in is all virological data, which I found interesting in a model. So they put there, as you can say, the ability to mutate and adapt. Now that is a kind of new thing, which I come back later on. The second thing they gave a lot of weight was, as you can see, the impact of those mutations on virulence and transmission. That was, particularly in flu, a big debate, which I will come back later. If you're allowed in your lab to mimic those mutations till the moment it actually spreads, for instance, among mammals, let alone among humans. You cannot do that among humans, but maybe among monkeys or whatever you want, which is gain of function kind of experiments, which were under a moratorium for some time. And I was involved at the time with when the editors of Science called me up, and I can give you that data. But it is important. Do we know how much impact how many mutations have? Now, you have to realize that, for instance, in live polio vaccine of Sabin, if you get vaccinated with that, and that was an experiment which one of the big early polio scientists did, Phil Miner, with his own kids. And what he saw is that the mutations that occurred during actually the vaccination, this is a live vaccine, that came very close to actually the virulence mutation that actually gives you polio and human-to-human transmission, which was quite a variance. And there it stopped. So that was an interesting learning experience. But anyway, that is an important point. And the other ones are transmissibility, of course, we talk about it. And what I really liked is the presence of asymptomatic transmission, because that is a very important one. The first thing I remember in a discussion here in 2020 with Barry Bloom was that we were discussing at a dinner. He was only saying, we only have to know one thing. What happens? Is there an incubation time at which there is a lot of transmission at a certain level in the asymptomatic incubation period? Because then you don't see it and it's difficult to fight. Like, don't forget that in smallpox, why is smallpox eradicated? Because of the fact that immediately when somebody is infectious, you can see it. Now, that is a helpful sign. You can isolate a person with pox. Now, which kind of viruses then do you have to think about? There were a lot of articles on that. This is another article which I find very interesting. What they put is, they put 12,000 viruses in their model. And they said, okay, what is the most important correlate of a virus that actually spreads among multiple mammalian species? Now, and what they came up with, this was two Spanish researchers, and they said the most logical thing and the most important thing is it's particularly enveloped viruses that do that. And they took as only measurement how many mammals species get actually affected as a measure. So here you have now a model where they used particularly a set of virological criteria, as well as, in this case, what kind of viruses are there. Now, if you take the most, out of those two models, the most important features of a virus, and you take that both into account, you get 3 questions left, which is what is the transmission route. Now, everybody understands that the respiratory route is the most effective one, and that is maybe relevant to think about. The highest transmissibility of a virus, in other words, that spread from one person to as many as possible, the most dangerous one is, of course, measles, which is not a new virus at all. But we have to think a little bit in those terms. And the transmission route is, of course, respiratory with measles as well as COVID and flu. So the key question then is which one is going to occur. Then the mutability is important. How fast does it change? And how does it change? And the capability of reassortment, which is typically for flu. Now, what I just put at the right side is a little bit which kind of viruses then pop up as enveloped, in this case mainly RNA viruses. And what you see is, you see already occurring, but that is hindsight. It's all talking about hindsight. That's the problem a little bit with preparedness, is that you see flu shows up, corona shows up, HIV shows up, which is quite amazing, because it's the same family of viruses, and Lassa and Ebola. And this is a little bit the idea which came up over the last 2-3 years as a kind of criterion on virological viruses to particularly look at. This introduction shows you how you could get at characteristics which have the highest chance. It doesn't mean that it's going to happen. Now, the biggest problem in preparedness is a little bit, what is that? It's a kind of preventing something you don't know it's going to happen, and it is heavily logistics. What I saw after both the AIDS epidemic, but we saw it also after the flu 1976, we saw it after the Mexican flu, that everybody talks about preparedness. The funding of governments of preparedness halves per year. So this is crazy. So it means the first year after a pandemic, there is a lot of funding generally available for pandemic preparedness. The second year, it's halved. And then in about half a decade to a decade, it's totally gone. Because it takes so much infrastructure to prepare for not happening. You have to realize if you start a factory, and I worked for 15 years in industry, if you start a factory, and you have to train people, you have to inspect the factory constantly, you have to actually exercise it constantly. It's so unaffordable that you have to find very intelligent mechanisms to go there. Now then I discussed a little bit, if we then focus a little bit on flu, there are 2 interesting aspects on flu. First of all, don't forget that in 1996, H5N1, everybody is talking about that now, was for the first time discovered in Southeast Asia. And what was interesting is I put a few papers up, but the most important one is here at the right side, airborne transmission of influenza virus between ferrets, and the one above that. And what is so important is the Science article. That was the big debate. What this group and the group of Kawaoka in Japan both did was an experiment. They took the virus and passaged it live. It's a very easy experiment, it's not cheap, among ferrets. And what actually made the virus more and more transmissible. And that experiment, which was done a very long time ago, suggested that at least we know a little bit the mutations from animal to animal and within certain animal species. We still don't know which mutations are vital for human-to-human transmission in flu, and we don't know how to actually get at that, and if it will be flu. Now, to confuse you a little bit more is lessons from the past. And lessons from the past I have became very skeptical about. In 2003, I was involved very much in making vaccines and antibodies against SARS. SARS occurred in Hong Kong in 2003. And SARS-CoV-1 is very close phylogenetically to SARS-CoV-2. Extremely close. We have to realize that. The next problem, the next issue is it's virtually immutable. It's virtually immutable. Second thing, it gives the highest infectivity is at the moment of symptoms. And the third one is the transmission route is mainly contact. Now this is a virus which is extremely close. It uses the same entry in the cell, ACE2, and we took from that, and it was very easy for us, and I was involved in that, to make antibodies to it, to make a vaccine against it. Very easy. So we thought when SARS-CoV-2 came around, it was easy again. This virus just vanished. God knows where it is now. We have no clue in which animal it is. It's just gone. We know it had an intermediate host, which was a civet cat, which was actually eaten at markets. So for that one we knew. Then occurred in 2012 MERS. MERS is even crazier. It's a virus which is also very close to COVID. However, it occurs year after year in Saudi Arabia and all of those in camel handlers. It stops at infecting camel handlers. It's 80%, 70% infectious, and it is also very deadly. So it occurs in 50 of those, and it sticks to that surrounding without us doing anything. So you had two experiences, and MERS vaccines are also easy to make, and it also doesn't change much. So I must say, personally, I was totally misled from the beginning. I thought it was SARS-like or MERS-like, and so the problem of pandemic preparedness here is we don't know. We really don't know. Even if it is that close of a virus, that close as you can imagine. So now recently there was a new one, which is HK, and in the end of the day, it's one of those problems is, can we predict anything? It's a little bit maybe crazy to say, but is it more important than children's vaccination, which is old technology and just, so let's discuss that at the end a little bit. What are priorities if we talk about public health? Now, to give you a little bit, this was the Hong Kong situation. It was an immutable virus. It behaved very nicely. And I spent a hell of a lot of time on finding antibodies at the time. It took us about a year to get. And there was a time that there was no sequence. I had only, the virus was isolated from a Singaporean individual in Frankfurt. So we said, okay, we're going to inactivate the virus and then select antibodies on the inactivated virus, which was an antibody called 3022 against SARS-CoV-1. And that was an antibody, I will not talk about too long about that, but something very important happened. We worked at and I was at the time already working in industry, and we made antibodies to it. We thought we have the therapy, very nice, and then it vanished completely. And that made us very nervous because we had a vaccine, we had an antibody, everything ready to go. And then the virus disappeared. So in other words, that is not really an incentive to do that again. It's not a really market, so to speak. Now, the situation for SARS-CoV-2 is also very interesting because at the early moment, a lot of discussions were on where does this virus come from. And I showed you already, we know a little bit where all these viruses come from. They generally come from a spillover in the animal world. However, there the buck stops, more or less, because we don't know how the selection occurs from getting to human transmission. Now, another experience was very important, how to go about it. And all our experience, particularly of young people, are in emergency vaccines. I was on a BARDA grant for an Ebola vaccine for the Army in the United States. We made that vaccine we made an emergency vaccine a little bit like we made the COVID vaccines, only with different technology. In that case, they were adenovectors. Now, of course, mRNA became very popular because that's so much faster to make. So the criterion was, how fast can we make a vaccine? mRNA is the fastest possible. It took us at least a year. But I wanted to tell you a little bit about how you can see that. What does an emergency vaccine do? It has generally a fast schedule. Now, that means that the primary immune response immunologically is mainly priming. You get a very high spike of protective response, but it wanes relatively fast. You don't care because you vaccinate everybody the fastest possible. The problem with that is also an experience we lost a little bit for follow-up. 1976, the swine flu. 40 million Americans were vaccinated in that year, and the pandemic didn't show up. It's a very important point. So the only thing you saw was side effects of the vaccine. And for the older ones among us, we know that then Guillain-Barré was one of the side effects of that particular vaccine. So everybody was talking about the danger of vaccines already in the 70s. And it was a massive government activity. I mean, you saw these pictures of Gerald Ford, I think, at the time, getting vaccinated, and there was an enormous amount of government money in that. Now, routine vaccination, that's are the vaccines we truly know what they're doing. These are the children's vaccines, are generally slow. They're long-lasting immunity, which had an enormous advantage, but they're mainly against viruses that actually are not mutating that fast, and they give this enormous high protection. Now, emergency vaccines is now what is almost became we started last week with the vaccine course, and almost all students only know emergency vaccines. It's kind of interesting, instead of the children's vaccination, which is much more important in public health. Now, if you then look how you go about that, and this is just the kind of vaccines I'm talking about in the last minutes, is we started therapy at the same time. Now, if you have a variable virus, this is a very complicated activity to do fast. You can actually find this antibody. Low-hanging fruit is easy to get. It took not even a year to make all these, and you see here the industry names of all of these antibodies that were made. The biggest problem was that all of these antibodies were targeting a variable region from the first mutant, which is the Wuhan strain, and we didn't count too much on how fast it moves into whatever realm. Now, I just want to point out how interesting epidemiology became from a virological point of view. These are actually maps, antigenic maps, of how the virus moved. And you have to realize that if you look at the variation moved so incredibly fast that we already could anticipate after a year or so that actually it would be very difficult to actually target variable regions of this particularly S protein, which was the outside of the coronavirus, to make a vaccine or an antibody against it. So here a real problem occurred. It actually meant that these drugs, these large molecules, are now all out of the market. All of them are gone because the virus mutated so much faster, and that is at the cost of billions. So in other words, billions were spent to actually make something that had a lasting period of maybe a year or two. So that is already a route which we now have learned that we should at least be suspicious about if you have to actually be or live in a time where money is relatively scarce. I spent not too long on this, but we also looked at what can antibodies actually do, and there is now the view also at the FDA and also at other regulatory agencies and health agencies that you're in trouble for the first 100 days. If you take the mRNA vaccine development, you know at a certain point the sequence. You can make an mRNA vaccine in 3 months. So in other words, you are left with a period in which you cannot do much, but do something nobody wants anymore, that is lockdowns or keeping people at home. Now the next problem was, which actually one of my PhD students pointed me out, and I'll show you that in a second, and I was discussing it with Bert the last time, is it turned out that the mRNA vaccines, the only ones we really understand well, induce essentially only a priming. And everybody said that's what these mRNA vaccines do, and I don't believe that. The mRNA are as good as any other vaccine, but the schedule is wrong. What they gave in the schedule is generally 0 and 21 days. Now, I grew up under the guidance of one scientist from Atlanta, from Emory, which was Rafi Ahmed, and he developed the idea of how do you get long-lasting memory. And long-lasting memory, you need to actually get a antigen to induce immunity much longer. If you take, for instance, the children's vaccines, you take, for instance, measles. Measles gives you, if you get vaccinated at young age, it gives you, that's research from Slivka, and there's a whole literature on it, it gives you lifetime immunity. But it is a long schedule. So if the problem occurs today, it needs time to do it, and you need the vaccine at that moment. So it's not feasible for outbreaks. This shows you how poor the immunity actually is. And everybody accounted the mRNA vaccines. I think it's technology independent, it's schedule dependent. But that's only recently that people start to develop that idea. We should have known that already. Now, one of my postdocs, I put her Nature Medicine paper up, actually pointed it out to me on the phone. And this was in 2020, in, say, after the summer. It was about midsummer. It was published in November. And it was based on an epidemiological study among homosexual men, which was already running for 30 years by 2020. Tells you a little bit about the importance of having samples, or importance of having... so what this prospective study started among homosexual men and IV drug users in the late 70s, I think, or when we started, 80s? 80s, early 80s. Yeah, early 80s, 83, 84, 85. And we sampled every half year in the end. Every half year we sampled serum and plasma at the time. And this study was using those samples, and she called me up and she said, it looks to me like this virus behaves like an ordinary common cold coronavirus. Now, there are 4 of those. Those 4, you and I get all infected with those yearly. Every year, one of those 4 seasonal ones. And she said, SARS-CoV-2 looks like, to me, like the beginning of how such a virus, all of these 4 viruses, have done this only 100 years ago. And then they started to become into humans, and then they spread among humans, and then they behave like this. So this virus might behave like that. She was completely right. But I thought, this is crazy. What she says is totally crazy. So in the end, everybody had, particularly in the United States, everybody had to reinvent, even after the paper, reinvent the wheel again. And it turns out that it looks like exactly now where we are going. So it's just learning how difficult it is to navigate knowledge at a certain point in a certain time. Now, a little bit about flu. The problem of COVID and flu is not that different, except that flu is much more unpredictable. Now, the evolution of the SARS-CoV-2 is pretty linear. In other words, the only really big break was Omicron, which occurred in South Africa for the first time. That is a strain of SARS-CoV-2, which occurred, and it was presumably, and not 100% sure, but I believe that, it occurred in somebody who was immune suppressed, so the virus could actually replicate for a long time in the same vessel, which was one person. And for at least a year, that's what they assumed, because that was the first person that was isolated from. And then it spreads and mutated from that new source. In flu, it's much more complex because it reassorts all the time. If you see how poor, this is from my own country, but there is from every country, the same data, that the flu vaccines are so incredibly poor for two main reasons. One of the reasons is mismatch. We cannot predict very well which one will hit and how severe it will hit and what is the primary virus. And the second problem is really is the age of the individuals. The more severe, the older you are, the more severe it is. So vaccine coverage is important, but it doesn't help you much to think through what you do in a pandemic or of any sort. If you looked at the very bad last flu season, you see how bad it is and how we really need better vaccines. And that is, I think, the good thing, at least of a lot of the previous 5 years, is there is a lot more research going on in finding a universal vaccine, which actually covers all of the strains. And I think it will be extremely hard to do. I worked for 15 years on trying to find that, and it was a total failure. I mean, every time, every experiment really failed. And just a little bit of that landscape, which one of our papers is at the right, which occurred reasonably much time in one of the researchers in my lab, Katrina Radosevich. We thought there was an immunological problem that if we finally had and it starts really with an antibody, I will show you, if we could make an antigen that could induce in a high enough amount of your B cells a set of antibodies that covered all of these strains, then you would be there. Now, this goal we never reached, and nobody has reached to date. Now, the other question we had is, of course, what happens with the seasonal flu vaccine? Does it induce these universal antibodies at all? And I think there is now a lot of agreement. We did an experiment in humans where we actually isolated, we took the plasma of a lot of individuals that we vaccinated with the seasonal vaccine, and we were looking if an H5N1 antibody occurred. And this was a publication from a few years ago. And essentially what we observed, if we followed these people for a long time before and after vaccination, which is in this direction, you saw that essentially in a very small percentage, yes, they occur, these universal antibodies, but they get deleted out of the repertoire relatively fast. Now that is interesting because it essentially means that the normal vaccines will not work against it. Now, if you go from all of these experiences, the question becomes, is there any way or any useful way of prepare yourself for something you don't know where it's coming from and what it actually is that is coming? And particularly if it is rare. Now, and so I give you 5 minutes of my own research, and that is essentially, I discovered in 2010, I think around that time, no, earlier, in 2007, after 7 years of research, one single antibody, which actually did everything. That antibody was so deep in the repertoire that you had to actually screen billions of cells from a huge amount of people. We finally found one, and that antibody is never found before and never found after we actually discovered it. But I can give it to you, and then you can agree or disagree. But that is an interesting point, because if you look at that antibody, and I will go through this, why it works, is we published as a company the sequence immediately after we discovered it and published it in Science. And this is just to show you a little bit what a huge group, this is data from 30 laboratories, which we didn't select at all. These were 30 laboratories, but there worked about, at the moment, 100 laboratories with this antibody. All of them showed that it neutralized every single strain except a few, like H2 and B strains. However, it protected to every single strain you used if you then did the protection experiment. And everybody now agrees that that is the case. Now, that means that then you have only one question left, that is this antibody useful than to give to everybody and bring to a market or make it available for everybody. Then because so many groups worked on it, I learned 2 lessons. If you have, and it depends a little bit how regulators look at it, but essentially the question is, not many research, I think not even more than 1%, even if it is extremely good, goes into benefit for people. Sometimes it does, sometimes it doesn't. For instance, this antibody nobody wanted to develop at the point that when I worked at J&J. There was no interest in it. But a lot of academic groups worked on it, and this is just an example of it. The top shows again all these 30 laboratories, what kind of neutralization data they got. And nobody got above this top non-neutralizing level, except one set of H2 and B. Then we used those virus challenge experiments to show that it was still protective through another function, which was Fc functionality. So it was 100% protective against all of these strains. The question was, of course, can this antibody be translated into and capture an antigen that actually makes a vaccine, and you can make this antibody dominant instead of recessive, because if you vaccinate, then you need a very dominant response to actually cover more than one virus. And it turns out a whole set of interesting points. First of all, it's very difficult with this antibody, which is against the fusion mechanism, which is essential for the virus. You cannot create escape. It took so many passages, we couldn't get it. It is essential for the virus. So as soon as the virus escapes, you cannot replicate the virus anymore. It just falls dead on its face. Now, we didn't even do that experiment. That was a set of other groups who did that. And also, for instance, the resistance you could create resistant mutants by that they didn't bind anymore. That was a lab experiment. But then that virus never occurred in nature so far. So in other words, over the last 50 years of sequences. So in other words, it's hard to find that. Now, so I stopped all my own research on a universal vaccine at a certain point because we couldn't do it. We actually made small molecules out of this as well, and we are now in the clinic with one of those antibodies. So if I finalize what to do is the first one lesson, which I find enormously learning, is that if you go for variable low-hanging fruit antibodies, then you lose the fight if it's a mutable virus. It was proven in the COVID pandemic. The second thing is, if you want to make a universal vaccine, the body doesn't want to make that. That's how it looks like. It looks like that if you do that, it only likes mainly to make, and it was very interesting, if you take the booster vaccine of currently COVID, you get mainly a rise in antibodies to the Wuhan strain, which was called original antigenic sin. And it looks like that really exists, which I never believed, but I think now that it is really true. Now then the other lesson I had was the lesson of failure. I'm an expert in failing, and the more experiments you do that fail, you have a chance to get success at a certain point. So never be afraid for failure. And this was an enormous failure. This was an antibody, it was on an NIH grant, was one of my NIH grants, and we showed that this antibody, this was a precursor of that universal antibody, this antibody proved excellent protection in ferrets, in mice, in all kinds of rodents. But then finally, it failed totally in a human challenge experiment because of our anatomy. It turns out that the antibody in plasma, this was delivered by plasma delivery, essentially IV. Then we saw almost nothing in the nose, where the affection... I summarize. Sorry, I summarize. Anyway, the bottom line of that is you have to put the antibody in the nose. Then it starts to look working. So you get this picture. All the failed antibodies have almost no antibodies in the nose. So we forgot that we are human, and you cannot actually... So finally we got it right, but it took us 10 years to get there. So I will go through it very fast. So the bottom line is a little bit, two lessons. One lesson is a pandemic preparedness drug or a pandemic preparedness vaccine should be licensed for normal, ordinary use, otherwise you don't have logistics. There's no way, in my opinion, that you can get it working for just pandemic preparedness because you don't know where to prepare for. The second issue is you need the first 100 days, if you have to make a new vaccine, you need a universal drug to actually give to people who actually, and that's another COVID lesson. Don't forget that the vaccines against COVID never worked against infection, like children's vaccines. Didn't do anything against infection. It spread like hell, still does. However, it worked very well to keep people at risk out of the IC. So you still need that vaccine. And so the bottom line is we have to be modest.